Hello, welcome to the 24th of June, 2021 MycoTalks. My name is Sarah Gaffen and I'm co-chairing this session with my colleague Iliev from Cornell University. I'm from the University of Pittsburgh. We are delighted today on this 12th MycoTalks series to have Dr. Julian Naglik and Dr. Elaine Bignell, both from the UK, talking to us today. A little bit about the MycoTalks, if you haven't heard them before. This was put together in light of the pandemic in order to bring medical mycology research to the broader community worldwide. The organizing committee is listed below, and this is sponsored by the MRC Center at the University of Exeter. And before I go any further, I really want to thank the University of Exeter and the folks behind the scene who are making this webinar work as smoothly and seamlessly as it does. These talks were put together, as I said, in light of the pandemic, and the goals are to promote diversity in both research field, but also career stage, gender, geography, to bring the world of medical mycology to the broader community. These are established as two 30-minute talks, as I said, covering various aspects of the field, which will be followed by 30 minutes of in-depth discussion that will be co-chaired by myself and Dr. Iliev. If you wish to submit questions, and we very much encourage that you do, please do this through the Q&A function on your Zoom screen. The chat is not monitored, so don't use the chat button. Please use the Q&A, and the questions will only be seen by the moderators. If you can, please indicate to whom your question is addressed so that we can help direct that question to the appropriate person. If you have to leave early, or if you have a colleague who missed this, all of these talks are on YouTube. They are being recorded and they will be available on the YouTube channel. Every talk, you need to register separately for every talk, and our upcoming talks are listed here for July and August. And finally, if you're a trainee, there is an opportunity to present through the Medical Mycology Trainee Seminar series. And we encourage everyone to log into these and watch them, and you can see the link here. So without further ado, I'm going to stop sharing my screen and turn the podium over to my co-chair, Dr. Iliev, who will introduce our first speaker.
Thank you, Sarah. It's a great pleasure to introduce our first speaker, Dr. Julian Naglik, who is a professor at the King's College in London. Dr. Naglik has had numerous contributions to the field of fungal pathogenesis, with specific focus on the human pathogen Candida albicans and its interactions with the host and the mucosal surfaces. His laboratory described the signaling pathways activated by the invasive hyphal form of Candida albicans, which in a recent collaborative effort was found to be due to the secretion of the peptide toxin Candida lysine. Professor Naglik has had numerous awards for his contribution to the field, and is a honorary member of the British Society of Medical Mycology. Julian, looking forward to your talk.
Thank you very much, Iliyan, Sarah, and the MycoTalks. I shall share my screen for you. Okay, hopefully you can all see that. Okay, so first of all, thank you, the chairs and MycoTalks for this wonderful invitation. It's been a fantastic series, and long may it continue. I have the title of Host-Candida Interactions, a Toxic Encounter, and of course this talk is about Candida lysine. So when I was asked to give this quite a while ago now, I didn't give it too much thought until about sort of 4 or 5 weeks ago. And I thought, because this Candida lysine story has sort of taken off a little bit, you know, it's not just our work, it's not just Bernard Huber's work, it's sort of a bit more of a global effort. Lots of people have been involved in it. So I wanted to sort of share the wealth in this talk, as it were. So I have to give you a bit of an introduction, which is our current status of where we are. I then want to talk a little about Candida Lysine localization. So the majority of this work has been done with Sergio Grünstein and his group, with Johannes Westman and Michelle Maxon, and with Bernie. I will then move on to looking at a slightly new model of epithelial signaling. So this might interest some of you. This is predominantly the work of our PhD student Spiridula Niku, who's at the Crick, together with Peter Parker. And we've done some work also with Sarah Gaffen on this. And finally, I want to introduce a new model, which you probably haven't heard of, which I think is a wonderful piece of work by David Corey's laboratory on how Candida Lysine might be driving asthma or allergic airway disease. So I wanted to introduce that if you didn't know about it already. So I've got a one-slide summary on what Candida Lysine does. So how is it activated? Well, it's activated through hyphal formation. So you get these hyphal-inducing signals. Candida makes hyphae. One of those genes is ECE1, which is part of the core response genes. It's been previously published by Bernie and others. This polypeptide is then processed by two kexins, KEX2 and KEX1. KEX2 cleaves after the arginine from this processing motif site, lysine, arginine, or KR, and then KEX1 removes the arginine to leave a K at the end. So all these peptides, or some of these peptides, are secreted, and Candida Lysine is this third one, and this is the sequence of Candida Lysine. And here you can see that the relevant references for the different parts of this processing work was done a few years ago. The original description of Candida Lysine was in 2016. So once Candida Lysine is secreted, this peptide, it intercalates into the membrane. It then, of course, damages the membrane. And as a result of damage, you get alarmins that get released, which we published quite recently, and also very recently, internal cell stresses, which include calcium influx, ROS, and intracellular ATP. We then know that matrix metalloproteinases are activated. These cleave your EGFR ligands, which then bind EGFR. This was published a couple of years ago. And then we have MAP kinase signaling. It's more than just MAP kinase, but this was originally identified about 11 years ago now. So this really needs a bit of updating. We've had lots of evidence since then to suggest that EGFR can be bypassed through this pore formation and damage. And also even the AP1 transcription factors can maybe be bypassed. So I'll get onto that in a second. So this is the current state of play, and this is oral epithelial cells, although it's very similar in vaginal epithelial cells. And these all lead to the release of pro-inflammatory cytokines. So that's the current status. So I'll move on to this part, which is Candida Lysine. So we know Candida Lysine is secreted, but exactly where does it go? What does it do? So this is, as I said, some lovely work from Sergio Grünstein's lab, and I'm just going to introduce 2 or 3 slides on this. So this is a nice summary of our work by Aaron Mitchell, which was the original discovery of Candida Lysine. So we know hyphae important. There's lots of other important factors of hyphae in addition to Candida Lysine. So you have HWP1, ALS3, SSA1. These are all important for adhesion, invasion, and to induce what we call this invasion pocket. In this invasion pocket, the hypothesis was that Candida Lysine is secreted. It then forms these pores and activates the cell. So we wanted to test this. So together with QVQ, with Edward Dolk and with Bernie, they created a camelid nanobody, a VHH camelid, and labeled it with Alexafort 88. And you can see in this nice sort of immuno, Candida hyphae penetrating into the cell, and Candida Lysine is labeled in this invasion pocket by this antibody. So the next question was, can you also detect it in an in vivo infection? So we got some human oral biopsies from oral candidiasis patients, and here you can see one of those. So the dark purple squiggles here are the Candida hyphae, and you can see that these Candida hyphae are darkly stained with this camelid antibody. So we can actually now detect Candida Lysine secretion in vivo. We then wanted to know, well, as it's secreted into the pocket, does it actually enter the epithelial cell, or does it stay on the surface? And this is some lovely sort of immunostaining suggesting that Candida Lysine actually stays on the surface of the epithelial cell, as you can see here, again labeled with this camelid antibody with 488. This is the nucleus stained with perdibin iodide because the cells are dying, they're lytic, and just connate to label inside. And when you do this central XY slice, you can see this is the membrane. Candida lysine is on the membrane, but it doesn't seem to get into the cell. So all those signaling mechanisms that I showed you earlier on, it seems to be doing it from the surface. Candida lysine doesn't seem to be entering the cell. So once it's in the invasion pocket, what does it do? How is it damaging the epithelial cell? So here you have a Candida hyphae on the outside. This is the point of entry. It's now on the inside. And at the terminal ends of this hypha, you can see this epithelial membrane labeled with FM464. It's a membrane label. And you can see these blebs being induced. And these are due to Candida lysine activity. So the cell is removing these damaged membranes. And if you do exactly the same with an EEC1 null, you don't see any blebs at all. So this is Candida lysine dependent here. And you can see very nicely that this FM464 label follows the hypha, suggesting that the membrane follows the hypha in this invasion pocket. So does Candida lysine do exactly the same thing? So if you just add synthetic Candida lysine exogenously, you can see all these membrane blebs being formed. This is a top view on top of the cell. If you do a side view, so this is inside the cell, and these are the surface blebs that are being thrown out by the epithelial cell, which probably most likely contain Candida lysine. I'll show you some data on that in a minute. So I just have to remove this laser to show you this. So we then wanted to see what's the effect on the cell. And we know that calcium is important. We've sort of demonstrated this before and suggested this. But if you label the cell with GCAMP6S, and then you add Candida lysine, this is a sort of fast forward of 15 minutes, you can see that you get these calcium flashes due to Candida lysine activity. So this is calcium flooding into the cell. It's then being chucked out of the cell, the blebs, as you see on the left, and then you get a reactivation through Candida lysine activity again. And this is a similar kind of video, but now you can see a bleb being formed where the arrow shows. So here you'll see an arrow coming up and a real-time bleb being formed in response to Candida lysine activity. I'll get mine. Okay, so what else does it do? So it induces calcium fluxes, we know this. We also know that it totally affects the mitochondrial membrane potential. So again, you have mitochondria. These are all these lovely squiggles. You have hundreds of mitochondria within your cell, and these are all nice and punctate and look like little worms in a vehicle control. But when you add Candida lysine, you can see that the membrane potential totally disappears and the rhodamine dissipates. So you get this very blurry green effect in the cell. So we know that the membrane potential has been destroyed. You can also show this with wild type hyphae. So this is a hypha going through a cell. I hope you can see in this outlined box that you get these fuzzy sort of rhodamine dissipation around the hypha, where you get these nice punctate mitochondria away. And again, you don't get this with the EC1 null. You get these nice punctate mitochondria, no effect on the mitochondria at all with the EC1 null penetrating through the epithelium. So how does the epithelial cell repair itself? So we know it damages. Does it repair itself? And the answer is yes. And there are two repair mechanisms that Sergio's team has found, which are very nice. One's called escort ALS2-ALEX system, and the other one is the Lysosomal Fusion system. So the first thing to show was, with these membrane blebs that are thrown out, is Candida lysine present in those blebs to demonstrate it's actually removing the damaged membrane. And that's shown here. The plasma membrane, you can see, is throwing out these blebs, and you can stain with Candida lysine in exactly the same area. So the cell senses the Candida lysine inducing pores and then throws it out. And this system is this escort ALS2-ALEX. ALS2 and ALEX are calcium sensitive. So as soon as Candida lysine makes this pore, you get calcium influx into the cell. These then get recruited and pinch this bleb off. So these are little blebs, and then fixes it. And you can see that in this schematic here. You get this pinching off the bleb and repair of the wound. However, there's a secondary mechanism, which is lysosomal fusion. And this one is due to mechanical stress. And you get this with long hyphae, not with short hyphae. And this one looks like it's Candida lysine independent mechanism. So here you can see a long hypha penetrating through the cell. There's no action at the beginning, but later on, you can see in the EC1 null, lots of distinct insertion of LAMP1 into the lining of these long invasion pockets, and you get exactly the same with wild type hyphae. So this suggests that these hyphae are penetrating through the invasion pocket. The cell can cope with some length of hyphae, but eventually they start tearing because of the length of the hyphal growth. And then this damage has to be repaired through lysosomal fusion. And that's just shown in this schematic here. These lysosomes come in to repair this damaged membrane. So on to the new signaling model work that Spiri Dula has been doing for the last 3 or 4 years as part of her PhD. So this is back to the signaling pathway that I was showing you. So I'm just not going to go through it again. What I want to target is all this here. So what's actually happening? Originally, we found this in 2010. There's been small little updates from various groups on this, including from Scott Filler and Mark Svidegal, some lovely work they've published. But we also know that there's some gaps. So rather than looking at Candida albicans here, which we've done before, we wanted to look at pure toxin. So if you add pure toxin onto epithelial cells, what are the signaling pathways and how do they interact with each other? So that's what we wanted to do. That's what Spiri wanted to find out. So just to say, this is an awful lot of work. You can see lots of different inhibitors that she used, lots of sRNAs, all the different signaling molecules. And this isn't all of them, this is just some. But I can't show you all this data because there's dozens, if not hundreds of Westerns. I'm just going to give you the punchline findings of the different pathways in a view of time. So there are 2 distinct pathways, P38 and ERK, and that's still correct. But we also know that there's some changes to what we originally thought. So pathway 1, Candida albicans makes Candida lysine poor. This induces EGFR ligands and binds EGFR. That's all correct, and that's all been confirmed with this model. EGFR then activates ERK1/2, and it's ERK1/2 that activates CFOS. If you remember originally in my previous talks, we always thought it was P38 that was the key driver of CFOS, but it doesn't seem to be. It's actually ERK1/2, and they're different, as in the P38 and the ERK pathways are totally distinct. And just to show you that, this is the only Western I'm showing you out of the 100 I was mentioning. So here you have your Verb inhibitor. So your Verb inhibitor here is inhibits all P38 isoforms, has no effect whatsoever on ERK1/2, CFOS, or MKP1. I'm not touching on MKP1 today. It's a regulator, a phosphatase regulator, but I'm just going to take it out of the equation for today. And when you use trametinib, which is a MEK1/2 ERK inhibitor, you can see total abolishment of CFOS and MKP1. So this tells us that ERK1/2 and CFOS, we've got all the EGFR inhibitor data as well, which confirms that it's EGFR that controls ERK1/2 as well, but I'm not going to go into that. Okay, so that's the EGFR ERK1/2 CFOS pathway. What about the P38 pathway? Well, we found that there are 2 pathways that activate P38. So this pathway, we found that P38 is actually upstream of EGFR and can phosphorylate it. Originally, we thought P38 was downstream of EGFR, but that's not the case. It seems to be upstream and can phosphorylate EGFR. And this requires Sark family kinases, and specifically Sark. So Candida lysine poor induces Sark P38. We think these 2 proteins interact directly, and then this phosphorylates EGFR. However, there's another P38 pathway, which is totally independent of Sark, and this is mediated by MKK3 and 6. This activates or phosphorylates P38 and a very well-known downstream target of P38. And together, this pathway, this MK3 P38 pathway, and this EGFR ERK-CFOS pathway seem to be the predominant pathways that control GM-CSF and GCSF. We were always fascinated a little bit by IL-6 because IL-6 was quite different. And what we found that we didn't really find anything with IL-6 with all these inhibitors. But what we did find is that P38 does seem to control IL-6 on the post-translational level through elongation factor 4E here. So we can reduce IL-6 by blocking this arm of the pathway. We did, of course, look at EphA2 phosphorylation from the work of Scott and Mark, and we found no effect at all on EphA2. We didn't do too much on this, but if you block P38, there's no effect on EphA2 phosphorylation, but you do find an effect on EGFR. So we think that these pathways, at least, are EphA2 independent. So then we moved into a murine model. So this is when we collaborated with Sarah Gaffen. You've probably heard her talk in the previous MycoTalk and seen many of her publications. But what we were a little bit disappointed in that we didn't actually find a major phenotype for our CFOS knockout mice. So just to remind you, the oral epithelium has two main layers. There's supraepithelial layer, which is K13 positive, and this basal epithelial layer, which is K14 positive. And you can use K13, K14 crees and cross them with CFOS flox mice to create your conditional knockouts in one layer or the other, which is what Sarah did. In a normal infection, so this is a wild-type mouse, Candida albicans 24 hours OPC, you can see that normal CFOS expression is in the K14 layer, as it is here, and then you get induction in the K13 layer in a wild-type infection. But in the K13 knockout mouse, you can see that you only retain CFOS expression in the K14 layer. You don't see it in this K13 layer. So this suggests that everything was great. The mice are working fine. So we continued with the experiment. However, a little bit disappointingly, we didn't actually see a lot. So this is weight loss. So the wild type mice and the CFOS knockout mouse, we saw very little difference in terms of weight loss, which you do see, of course, with previously published mice, the ACTA and the IL-17 receptor with IL-17 signaling. We also looked at CFUs, and again, the mice quite happily clear the fungus, just like wild type fungus. So there's no effect on CFUs, but you do, of course, see that with your control IL-17 knockouts and ACTA mice. So what about the Verb inhibitions? This is P38. If you recall, Verb inhibits all the P38 isoforms. Here we did find an effect. And this is in terms of CFU, I'm just showing you here. So at day one, there is a significant increase in CFU when you inhibit P38, and this increases further at day two. So this tells us that P38 is important for this early clearance of Candida. However, by day 4, everything's recovered and the Candida gets cleared. We then decided to look at some of the cytokines involved at these day 1 and day 2, and this is where we had slightly disappointing results in that we didn't see huge changes in the mRNA of the numerous genes. I've listed a few of your favorites here maybe, so I won't go through them. So we know that P38 is involved in this early clearance, but we don't know how yet. So on to the final part of this talk, which is the main part. So about 10 or so minutes to finish off. And this is the in vivo aspect. So we know Candida lysine is important for many in vivo infection models. We've shown this before and published it, so I don't want to reiterate too much about that. But I do want to give you some summary slides of what's happening in different sites, because I think that is important. And I'll focus at the end on how it might be driving allergic airway disease or asthma. So Sarah has published many on this, as have so many others. We've published together on the oral model. We've published with Brian and Scott on the vagina systemic model, and Michalis and Rebecca on the brain meningitis model. And in each, these are the relevant references that you might want to go to read about them. In each of these models, we found that Candida lysine does very similar things in that it induces damage because it's a pore former, it induces inflammatory responses, and neutrophils seem to be key in terms of recruitment and protection. So when David Corey approached us to look at the role of Candida lysine in asthma, we were highly intrigued because we're not asthma clinicians or academics, and so we're very intrigued to see what we might happen in asthma. And this work has just been submitted. And so David was very happy that I could present some of this with you today. What I'm going to do is just quickly summarize what's happening orally, vaginally, and or systemic in the brain, and then move on to the asthma. So what happens in oral infection? I've just shown you this. This is the new model. So we know that the cytokines I'm not going to go through it again, get released. However, we also know that many other cytokines and chemokines are secreted. For example, IL-1 alpha beta. So there's going to be other pathways that Candida lysine activates here that we don't quite know about yet. So these might account for some of the secretion of these cytokines. Irrespective, you have a secretion of a whole load of cytokines and chemokines due to Candida lysine activity, and these recruit certain cells. One of which we know, of course, is a neutrophil, and the other one, which is lots of work done by Sarah, Salome, and numerous others, are these innate types of 17 cells. I've listed a few here, and these then get activated in different ways neutrophils can get activated, secrete TNF-alpha and antimicrobial peptides, alpha defensins. We know that IL-1 receptor and IL-36 receptor seems to be important for these cells. They respond to IL-1 alpha and IL-1 beta, which bind to this receptor. You then get secretion of IL-17, IL-22. And the function of this, we think, is to upregulate antimicrobial peptides, other cytokines. Neutrophils can form nets. And we also know that Candida lysine can activate neutrophils. I'm not going to call them nets. We can maybe call them net-like structures, or we can potentially call them Candida lysine-induced nets. But they are not classic nets. And this is work we've been doing together with Konstantin Urban. And together, these neutrophils and innate T cells protect against infections. This is the oral model. So what about vaginal infections? Well, it's similar, but also very different. So this is a model of vaginal infection. We think this part is very similar. So how Candida lysine activates the vaginal epithelial cell, we think is similar, because you get a similar release of these cytokines, although there are some changes and some tweaks to this, which I won't go into now. However, it's been shown by Paul Fidel's group and a number of others that T cells aren't really, and these innate T cells aren't really important for vaginal infection. So if they're not important, you can probably rule out this potential pathway. However, you still have neutrophils. However, it is slightly different in vaginal infection. So those of you who know some neutrophils, it has a complement receptor 3, otherwise known as MAC1. This receptor will bind opsonized Candida, for example, which might be opsonized with C3b, and also bind to PRA1, which is a protein predominantly found on Candida hyphae. And this can then target the fungus. However, what is happening in women with vulvovaginal candidiasis is they might have high levels of heparin sulfate in the vaginal fluid. And we know that heparin sulfate can bind this receptor and block it. So if the neutrophils are recruited, but they don't seem to be activated, and therefore if you can't activate them, then you remove this arm of the protective response as well. So if both of these protective arms are gone, then Candida can proliferate, produce Candida lysine, and this is meant to be Candida lysine, and then induce damage. So this is what we think is happening, and this is what's driving immunopathology, we believe, although this needs to be double checked with patient samples. So what's happening in the brain system? Again, a slightly different system in the brain. So this is the lovely work of Rebecca and Mihalis. So these are my little baby Candida coming through the blood into the blood-brain barrier. And for those of you who didn't know, Candida can quite easily cross the blood-brain barrier. And here it proliferates, which is why it's getting larger. It makes Candida lysine, and then Candida lysine activates these card9 microglia. These card9 microglia make IL-1 beta, which activate the astrocyte, which then in turn can make CXCL1, and the microglia also make CXCL1. Candida lysine can also bypass the microglia and activate the astrocytes directly to release CXCL1. CXCL1 will then activate and recruit the CXCR2 expressing neutrophils, and it is these neutrophils that we believe protect and then deal with the fungal infection. So that's what's happening in the brain. So what about allergic disease for the last sort of 10 minutes or so? So not to upset the Aspergillus crowd too much, so I've put Candida almost as important in asthma as Aspergillus. It's quite an old publication now, but you can see the numbers are very similar, but Candida is second. So I'll move swiftly along to the actual model, which is here. So the model that David and many others use is every 2 days you apply Candida albicans 10 to the 5 intranasally, and then challenge at day 18 with acetylcholine, and you look at respiratory function. So this is a standard model. So we looked at Candida, and when you add wild type Candida, you can see the airway resistance is markedly up, suggesting a Candida albicans induced asthma or allergic airway disease, which you don't get with a PBS control. So the first question is this the same as Aspergillus asthma? So with Aspergillus, for those of you who study this, it's the proteinases fibrinogen and TLR4 seem to be very important for Aspergillus-driven asthma and allergic airway disease. So David looked at all of these in Candida and found that none of these were involved in the Candida response. It was at this stage we introduced Candida lysine to David, and he was very keen to test whether Candida lysine might be involved. and then the project came alive at that point. So here you have your wild type Candida, same model, airway resistance high, but with the EEC1 null. So if you knock the gene out, this is a Candida lysine or EEC1 null. We'll just call it a Candida lysine null for ease. You can see hugely reduced airway induced allergic disease. If you then look at the bowel samples from here, you can see total immune cells high, and these are predominantly in terms of eosinophil infiltration, which are markedly down with the EEC1 null, although you do get some MACs and neutrophils going down as well, which are recruited in the wild type. If you then look at the cytokines, so the Th2 cytokines here, IL-1 and IL-5, markedly increased with wild type Candida, markedly decreased with the Candida lysine null. And then the Th17 cytokine, IL-17, markedly increased with wild type, markedly decreased with the EEC null. And it wasn't all cytokines, because if you look at interferon gamma, for example, there's no effect between wild type or the EEC1 or Candida lysine null. You can then do some fax plots. I put this in for the immunologist. I know they like looking at fax plots. So this is whole lung sections, or looking at Th1, Th2, and Th17 cells through their transcription factors. You can see no effect here for Th1 cells, but markedly reduced Th2 and markedly reduced Th17 cells when you do the infection or the model with the EEC1 null. However, like we found with many of our other models, the CFU of the EEC1 is very high in the lungs. So you can see how much more CFU is present in the lungs with the EEC1 null. But so this tells you that EEC1 is not cleared, but it fails to induce allergic airway disease. So this is interesting. We found a similar thing with vaginal infections, for example, and with the brain model. This is just some histology. Don't want to dwell too much on this. This is wild type H&E and PAS. So lots of inflammatory cells in the alveoli, neutrophils and eosinophils mainly. Also, goblet cells are abundant, and this can be seen with PAS stains. And goblet cells are very important for airway remodeling and asthma. And you don't see this in the EEC1 null for the H&E or the PAS, and it's very similar to the PBS control, suggesting that it's the toxin that's inducing these effects. So that was albicans, you know, the actual fungus. So what if you just give them Candida lysine exogenously to the airway? Well, you find a very similar effect. So here's your control. Here you have different doses of Candida lysine, so 8 to 20 and 50. You can see airway resistance markedly increased, suggesting allergic airway disease is being initiated by Candida lysine alone. Again, if you look at the bowel cell count, you have this dose-dependent increase in total immune cells, MACs, and eosinophils. In this case, it's more MACs and eosinophils, but we think the eosinophil is probably more important. Again, looking at IL-1, dose-dependent increase in IL-17. At this stage, it was a case of what to focus on. We know Th17s are important, so this became less sort of exciting maybe, but what was slightly more exciting is what's happening with Th2 cells. We haven't really reported Candida lysine and Th2 cells before, so David was very interested in pursuing this. Now, this is quite a complicated slide, so to keep it simple, there are many activators of allergy, for example. So one mechanism is this Th2 cells. Just to point out, P38 is involved in this, and we know Candida lysine can directly activate P38. But another way of inducing allergic airway disease is to activate platelets. And this seems to be via a peptide called DKK1. And this has been shown to be very important, I believe, in leishmania allergy and house dust mite allergy. And then this can also drive like a type 2 chronic inflammation. So the question here is Candida lysine important? So David's group took mouse platelets and showed that wild type Candida induced DKK1 and Candida lysine induces DKK1. Same data was found with human platelets. And the question then is, if DKK1 is so important, if you add a DKK1 inhibitor, can you prevent airway disease? And the answer seems to be yes. This is your wild type Candida, and then the wild type Candida together with the DKK1 inhibitor. And just to remind you, this is very similar to the EEC1 null. So induces a little, but not much. So it looks like Candida lysine is certainly inducing DKK1, which is then promoting this allergic response. Again, looking at the bowel, massive decrease in your eosinophils when you add your DKK1 inhibitor in the dose-dependent fashion. So it seems that eosinophils are driving this. Again, some more facts plots. You can see that when you add DKK1 inhibitor and take the whole cell lungs, you can see a massive decrease in Th2 cells from 47 to 17. Likewise with your Th17 cells down to 8. And when you look again at the cytokines, massive decrease in IL-4, IL-5, IL-13, and IL-17 cytokines in the presence of the inhibitor. So the next one I wanted to show was if you add DKK1 to the EEC1 null, then surely that should promote the allergy, because we know the EEC1 doesn't, but DKK1 does. And again, that's what we find here. EEC1 null doesn't do too much. You add the DKK1 inhibitor, you drive allergy. Likewise, you drive eosinophilia. And again, you can see that you increase Th2 cytokines and you increase Th17 cytokines through the addition of DKK1. And this is just to show you the actual protein levels, massive increase in IL-1, 5, 13, and 17 in the presence of DKK1 when you add the EEC1 null. And again, just to show you the CFU, very high CFUs in the EEC1 null, but the DKK1 inhibitor reduces those CFUs. So you have this sort of opposite correlation that DKK1 drives asthma, but also removes the fungus. So on to the final 2 or 3 slides. How is Candida lysine doing it? Well, it seems to be binding this surface protein called GPP1 alpha. I'm not going to go through all the data, but David's group inhibited lots of platelet surface receptors and found GPP1 was the only one that seemed to be involved. You can pull Candida lysine down with GPP1 alpha. You can show that there's binding by flow cytometry, and you can block this binding with an anti-GPP1 antibody. And that's really just shown here in graphical format. You can block binding of Candida lysine to platelets here. That's a summary of these data sets. And you can also block activation of platelets. So activation marker of platelets is CD62, P-selectin, and you can see that you can block activation with an anti-GPP1 alpha antibody. This was all confirmed with controls, which had no effects. It was certainly GP1B alpha, and a natural ligand of GP1B alpha, which is von Willebrand factor, can also block Candida lysine binding. So it seems that Candida lysine and von Willebrand factor can compete maybe for the same binding site. And the final data slide before the acknowledgements is to show that platelets are critically important for homeostasis and protection. And that's just shown here. That if you do a similar model in a platelet deficient mouse, you can see if you give them wild type Candida, they die incredibly quickly, which is this red line, within 6 hours. So this isn't days, this is hours, but with the EEC1 null, it's no effect. So we know Candida lysine is driving this. But there's no effect if you directly administer Candida lysine, and there's no effect if you use platelet sufficient mice. So it has to be Candida lysine produced by the fungus in an infection. If you just quickly look at some histo, you can see the lung is full of blood. This is confirmed through histology, whereas the EEC1 null is quite normal. You can look at the bowel, it's full of blood, the EEC1 is not. And if you do a CT analysis of the mouse lungs, you can see that it's full of this dense regions, which we think is blood. So what's the model? This is just a quick summary of the model to summarize that. Candida gets into the lung, and then this is what's happening in the lung. It makes Candida lysine, binds GPP1 alpha on platelets. Platelets make DKK1 peptide, and this drives Th2 and Th17 allergy disease together with eosinophils. How this all works at the moment, we're unsure. At the same time, Candida albicans secretes Candida lysine, which can damage the endothelial membrane of the blood vessels. Normally, von Willebrand factor is induced, binds the same receptor, and leads to homeostasis, and the platelets block this. Whereas, of course, if you don't have platelets, hence that platelet deficient model, none of this protective factor can take place, and you end up with hemorrhaging and death. So I just want to say thank you to all the group, and I'm just out of time. This is the current group that we have, a fantastic set of people. I can't present all of their data. Hopefully in future talks and meetings, I can do this in person. just to highlight two very important people who helped us establish lots of the original data, David Moyes and Jonathan Richardson, lots of other colleagues. I'm just mentioning the 4 key people that I've induced the data of today, but we're actually collaborating with many people here. I'll just leave this up. It's all in alphabetical order based on surname. I apologize if I've missed anybody out. I haven't done it on purpose. And on that, I will finish and thank you very much.
Great, thank you very much, Julian. It is my pleasure to introduce our next speaker, Dr. Elaine Bignell now from the University of Exeter. Elaine did her bachelor's degree training at the University of East Anglia and her PhD in fungal genetics with Herbert Arndt at Imperial College London. She remained at Imperial College for a number of years, rising through the ranks to the position of lecturer. And in 2013, she relocated to the University of Manchester, where she became one of the founding members of the Manchester Fungal Infection Group. She left Manchester in 2020 to become the co-director of the MRC Center for Medical Mycology, which is where she is now. And Elaine is really a pioneer in the field of Aspergillus genomic, transcriptomic effects on pathogenicity. And she incorporates both genetic, fungal, mouse models, and human studies to really bring together an understanding of the molecular basis for pathogenicity. And a particular focus of hers has been on pH sensing, and a lot of both basic science and translational impact has come out of that work. And I think she's going to tell us about some of that today. So let me turn the podium over to Dr. Bignell. Thank you very much.
Thank you, Sarah. And I want to thank all of the organisers for a wonderful seminar series, but also the opportunity to speak. It's a privilege to do so and a privilege to be part of this wonderful research community. So let me see if I can share the screen. Jules, can you tell me, is that the presenter view you've got or the slide view?
That's perfect, Elaine, thank you.
Smashing. Okay, so I want to begin my talk by just pausing for a minute to think about where we're at really with human fungal infections. And when we think about infectious diseases, there are a number of important factors to consider. So one of these is how frequently these diseases occur, and another is how many of those infected will die from their disease. And the number of deaths occurring is the product of these two factors. And these are 3 metrics that we often use to measure how well we can tackle particular diseases. So last year, there were over 100 million SARS-CoV-2 infections that led to 1.8 million deaths, a case fatality rate of 1.8%. And this would seem to be a better management scenario than that of TB that caused a similar number of deaths last year, but affected far fewer people. And it's worse than malaria, where many more people were infected, but far fewer died. Deaths from invasive fungal infections also reach the millions in number annually, but on average, they kill 50% of those infected. And many of these deaths occur despite antifungal treatment, and mortality rates for drug-resistant disease approach that of Ebola, for which there is no known cure. So as the at-risk patient populations continue to diversify and expand, and rates of antifungal resistance increase, no stone must be left unturned in addressing this important threat. So Aspergillus species cause a range of fungal lung diseases in highly disparate clinical settings, and these diseases can be rapidly fatal, chronic or allergic in nature, and now include viral pneumonias as risk factors for acute infection. And across the spectrum of invasive and chronic disease, frontline therapy, including prophylaxis in neutropenic hosts, is reliant upon a single orally bioavailable drug series, the azoles. However, in recent years, the dual use of azoles as medicinal and agricultural fungicides has accelerated the emergence of clinical azole resistance. And we know this from the spatiotemporal trends in reporting of azole resistance, depicted here by Matt Fisher and colleagues as advancing rapidly across the globe. And on the right, data from a clinical study led by Paul Vevey that revealed voriconazole resistance to be associated with an excess mortality of 25% at day 90 in patients with invasive aspergillosis. In bacteria, the evolution and dissemination of antibiotic resistance has already outpaced the development of new antibiotics. And this has led to intensive interest in alternative approaches. And recent surveys indicate that screens for directly acting small molecule growth inhibitors now account for less than 50% of all discovery programs. And more than a quarter of new efforts seek antibodies, vaccines, or antivirulence drugs that instead focus on disarming pathogens by neutralizing their virulence factors. These approaches often circumvent the severe selective pressure that can be imposed by blocking essential cellular processes. And because these new approaches often use bespoke phenotypic screens, they offer new opportunities to screen existing chemical libraries under conditions that would not have previously been used. And the use of these agents as adjuvant treatments alongside existing antibiotics might promote the more judicious use of antibiotics or augment their efficacy. But all of these benefits have to be balanced against obvious drawbacks, such as redundancy or strain specificity of specific virulence factors, or the fact that these agents might not be effective for all forms of disease caused by the same pathogen. And because this point is so relevant for aspergilloses, I'm going to come back to it later on. So Aspergillus fumigatus is an important recycler of organic matter in the natural environment, and it accounts for the majority of human aspergilloses, but it's currently unknown how this fungus evolved to survive in the human lung. We do know that this fungus grows very rapidly at 37 degrees. So, for example, in less than a day in highly susceptible mice, inhaled spores can germinate and produce tissue-invading hyphae that can very quickly overcome residual host defences and decimate host tissues. And as an accidental pathogen, the most plausible antivirulence strategies might focus upon pathogen-specific processes that are indispensable for establishing infection. And those might include epithelial entry, melanin production, developmental growth of inhaled spores, or adhesion to host substrates. And in the disease progression stage, the adaptations that are made by the fungus to the host environment and which drive colonization and tissue damage, such as acquisition of micronutrients, metabolism, toxin production, and secreted protease expression could be useful targets. Now, many of the processes that drive disease progression are regulated in response to extracellular pH. And so in part one of this talk, I'm going to use fungal pH signaling to demonstrate how a deep understanding of the genetic regulatory circuits that drive Aspergillus pathogenicity can be exploited for drug discovery. And in part two, I'm going to talk about recent work that we've done to rapidly uncover all of the transcriptional regulators in Aspergillus fumigatus across highly disparate host pathologies that could be eligible targets for future drug discovery programs. So, fungal pH signaling. For all fungi, viability at extremes of pH requires genetic control of transporter and enzyme functions, because the fungal cell has to offset cation toxicity, maintain active uptake of essential micronutrients, and it has to safeguard other essential processes like cell wall biosynthesis from pH-mediated interference. And in Aspergillus species, the existence of a genetic locus that was called PAC-C that coordinated secreted phosphatase expression had been known since the 1950s. but it would be several decades more before its identity as a zinc finger DNA binding protein would become resolved. But in the intervening pre-genomic years, many upstream regulators of PAX-C were discovered via a genetic screen that made fungal growth reliant upon a PAX-C repressible gene function. And this was a gene that encoded a gamma aminobutyric acid uptake transporter permease called GABA that would only allow GABA utilization as a source of nutrients in the absence of PAX-C activation. And this was a very powerful genetic approach, and many hundreds of loss of function pH signaling mutations were selected for in this way. And then by physically mapping all of those mutations to their chromosomal locations, the involvement of 6 further PAL proteins, I, H, F, C, A, and B, was identified. So the presumed pH biosensor PAL-H is located in the plasma membrane, and via interaction with PAL-I and a cognate arrestin, PAL-F, it elicits the post-translational modification of PAL-F that leads to recruitment of the PAX-C transcription factor and various other ligated protein scaffolds, which are required to ultimately effect the cleavage of the transcription factor that becomes released from the complex for nuclear entry. So the PAL proteins are necessary for this processing event, but they're not sufficient on their own for PAX-C processing. And this requires also some co-opted members of the escort machinery that Julian spoke about in his talk. And these are shown here in yellow. Now, these proteins were not identified by genetic screens in Aspergillus because loss of function in Aspergilli makes Aspergillus strains virtually inviable. And instead, their involvement was uncovered in a series of very elegant studies from Aaron Mitchell's lab using Saccharomyces cerevisiae. So the proteolytic cleavage of PAX-C provides an excellent readout of signaling activation in Aspergilli. And this can be observed and quantified via mobility shift assays that reveal a preference for the short form of the transcription factor at alkaline pH. And in all genetic backgrounds that we've worked in so far in Aspergillus Fumigatus, this effect is observed. I'm showing you experiments in 2 genetic backgrounds here. And this is a response that obviously is absent in the absence of PAX-C, but which is completely abolished in the absence of the upstream PAL-H biosensor. And in laboratory culture, abolition of pH signaling results in alkaline sensitivity compared to progenitor isolates and also cation sensitivity. And in mice, both loss of the transcription factor and of the pH biosensor lead to severe attenuation for virulence, with inspection of infected tissue revealing a complete lack of invasive hyphae at a couple of days post-infection. So over the years, we've performed a lot of time series analyses of the lab-grown and infecting germlings over a 3-day period of infection in mice. And this reveals a transcriptional program that involves more than 250 fungal gene products that may be secreted into host tissues because the putative translation products include a signal peptide sequence. And of these, just over a third are not expressed in host tissues when pH signaling is blocked. And this includes all members of the gliotoxin biosynthetic gene cluster. So infection of cultured pneumocytes with these pH signaling mutants results in a 60% reduction in pneumocyte monolayer integrity, because although the mutants generate hyphae in these assays, they fail to activate the host response pathways that are important for driving lytic death of the host cells. And finally, the predictions of the in vivo transcriptome data are borne out by in vitro analyses. So, for example, the loss of pH signaling leads to complete abrogation of mucin degradation. These are data for 2 of the major mucins, and this is degradation of recombinant mucin. And also there is a complete loss of gliotoxin biosynthesis, and related to the dysregulation of cell wall enzymes, the pH signaling mutants are far less tolerant to echinocandin exposure, and that's true both in the lab and also in mice. So all of these in vitro and in vivo studies demonstrate that in Fumigatus, the fungus-specific Ph signaling mechanism occupies a central role in regulatory governance of tissue damage, degradation of extracellular matrix components, toxin biosynthesis, and cell wall integrity and pathogenicity. And therefore it fulfills many of the criteria expected of a target for antivirulence strategies, but without falling prey to this conundrum about redundancy of virulence factors. So in a final series of experiments, we asked whether blockade of pH signaling elicited in established infectious lesions could resolve acute lung infection. And to do this, we introduced a doxycycline repressible allele of the pH biosensor into a PAL-H null background. And this enabled us to dial down expression of the biosensor upon addition of doxycycline to lab media or when added to the drinking water of infected mice. And in the lab, doxycycline led to a complete elimination of the PAL-H protein. So this was a HA tagged version of the protein that we could no longer detect in the presence of doxycycline, and it led to a reduction in alkaline and cation tolerance. And in the animals, when added from 24 hours post-infection, it led to a 60% reduction in infectious lesions in those animals. So by now, we've gotten to thinking a lot about how to block the pH signaling mechanism, and we reasoned that the 7 transmembrane domain pH biosensor might conform to the precedent that lots of other 7 transmembrane domain proteins do, and that is to exhibit highly druggable properties. And when Bethany McCann began an industry-sponsored PhD project in my lab, we spent many months of the first year of her PhD designing screening strategies that would allow us to prevent the interaction of this protein with downstream signaling components. So in effect, we were looking to bust that signaling complex apart. But a game-changing conversation took place in our first collaboration meeting with Dave Cook from Blueberry Therapeutics, who observed that our plans would likely not lead to an all or nothing readout on inhibitor screening platforms. And it was one of those stop and rethink everything moments where you go off and eat a knickerbocker glory and have a serious rethink and change in the thought process. And it then became apparent that the answer to the screening conundrum had in fact been there all along. And it lay with this PAX-C repressible essential gene function. So we went back and we revisited the genetic screening strategy that had performed so well in the late 90s to identify regulators of PAX-C signaling. And in Aspergillus nigilans, the GABA permease expression is maintained in the cell by the positive action of the INT-A transcription factor, which then competes with PAX-C for binding at the GABA promoter when PAX-C signaling is activated. So when PAX-C signaling is disrupted, INT-A is able to activate GABA expression the fungus grows. And this provides us with a very simple readout of pH signaling. When signaling is on, the fungus cannot grow, and when signaling is off, it can. So for our initial experiments, we worked with Aspergillus nigilans, where we had a whole host of existing mutants that were proficient or corrupted for pH signaling. And we showed that we could reliably distinguish between wild type strains in red here that are unable to utilize GABA and to grow, and the pH signaling interrupted strains in black that are. And this worked very well in 384 well format. So in proof of principle screening experiments, we took a fragment-based approach to seek commercially available chemical fragments that could inhibit this signaling pathway. And we used the most chemically diverse 500 compounds of the Maybridge library to seek molecules that could inhibit pH signaling. And this identified 10 fragments which promoted GABA utilization, and I'll focus on 2 of these in the following slides. But these data were very exciting to us because they demonstrated that we developed a cheap and simple whole cell genetic screen for chemical inhibitors of pH signaling. And to satisfy Dave Cook's concerns, it exhibits maximal signal to noise ratios. And it also, because it acts in a whole cell format, may help us to identify molecules that act at or near the cell surface, or that are proficiently internalized. That would therefore offset a lot of downstream work on trying to get inhibitors into the cell. A further plus is that because the screen is focused on growth of the fungus, we can automatically circumvent molecules that hit essential cellular machinery, such as the escort, vesicle trafficking components or proteasome components that are highly conserved in animals and plants and critical for cytokinesis, and so would therefore likely select for toxic molecules. And then the other plus is that because nigilans is much less pathogenic and the spores are less buoyant in the air, we can work very easily on the open lab bench or in multi-user settings without any safety or cross-contamination concerns. And we can also exploit parasexual genetics and all of the genetic tools to try to identify the targets of these molecules and their modes of action. So a few experiments that we conducted to look at whether or not these fragments were inhibiting PAXI-mediated pH signaling. These are the data from the previous slide that show that the chemical fragments 1 and 2 are able to phenocopy a PALH null mutant in vitro. If these molecules were really acting on the PAXI signaling pathway, then we'd expect this effect to not occur. Sorry, my slides are advancing on their own. We'd expect this effect to not occur in mutants that constitutively process the PAXI transcription factor. In fact, that's what we see. These molecules are unable to reverse the phenotype of GABA non-utilization in this setting. So we also took a much more targeted molecular approach, and we constructed a synthetic PAXI-dependent reporter gene using the promoter of the NO1 NA sodium ATPase that has 9 PAXI consensus DNA binding sequences, and we fused this to YFP to give us a readout, a molecular readout of PAXI proteolysis. And so in these data, I'm showing you that compounds 1 and 2 are able to significantly reduce YFP fluorescence in this screen to an amount that's equivalent to introducing the reporter into a PAXI null background. We were also able to show that these agents combine favorably with caspofungin that we expected from our genetic studies that I showed you earlier, but also with 5-flucytosine, the importer for which is regulated in a pH-dependent manner. These data show that the activity of our fragments is able to reverse the pH sensitivity of that antifungal mechanism. So drug discovery is an iterative process, and there are several design, make, test cycles that have to be conducted. This process involved 1,200 compounds in the search for voriconazole. But in our pilot studies conducted in partnership with Ed Griffin at Medchemica, we established a precedent for structure-activity relationships quite quickly via 3 iterations of the DMT cycle. And this used 7 off-the-shelf derivatives of our chemical fragment 2. And then we used the stringent in vitro pipeline, and we worked to, by a process of elimination, figure out which of the head groups on these molecules were important for antifungal activity. So these studies were pilot studies that were submitted to the BBSRC recently, and we've now secured substantive funding to take these studies further with bespoke syntheses and a much expanded screening programme going forwards. So in the last few minutes of my talk, I just want to focus on some recent work that we've conducted to perform the rapid identification of genetic regulators of pathogenicity in Aspergillus fumigatus. So this was work that was funded by the MRC and was conducted as a collaboration with Mike Bromley in Manchester. And at the time that we began these studies, there wasn't really an awful lot known about the role of transcription factors in promoting pathogenicity in Aspergillus fumigatus. So of 46 transcription factors that had been characterised to date in Aspergillus fumigatus, 37 had proven to be critical for survival in susceptible models of Aspergillus disease. But interestingly, although we know that Aspergillus affects highly disparate patient cohorts, we very rarely look at pathogenicity in more than one model of disease. And in light of the increasing urgency to counter the threat of azole-resistant Aspergillus fumigatus, we aimed to perform an experiment that would rapidly define the regulatory landscape of fumigatus pathogenicity by performing a genome scale census for transcriptional regulators that drive fitness during fungal infection. So for this, we combined the library of mutants that Mike Bromley's team had constructed and has already been documented last year with a high sensitivity, high throughput microfluidic based qPCR protocol. And we took a pooled infection approach to assess in-host fitness. And to identify regulators that drive colonisation in diverse host niches, the screen was performed in parallel using immunologically distinct mouse models, so myelosuppressive immune ablation or high-dose corticosteroid regimens that recapitulate the 2 most common clinical risk scenarios for pulmonary aspergillosis. We optimised the infectious dose of the fungus and the time scale of the disease in both immune models, and we conducted the experiment across a time course that involves the neutralising or the residual neutralising immune response. And we then were able to quantify the fitness of 481 individual Aspergillus fumigatus null mutants and 5 control isolates in 5 mice each for each of the 2 susceptible host models. And this yielded a data set of 4,810 individual fungal fitness assays performed in technical duplicate to yield a data set of more than 12,000 data points. So these are all of the outputs from the screen on a single plot, and here I'm plotting fitness of each mutant relative to a progenitor isolate, either in the corticosteroid model or in the leukopenic model of infection. And hierarchical clustering assigned these mutants to 6 clusters according to the magnitude of effect and their fitness phenotypes. I'm showing you here the data for the orange cluster, cluster 1, just to give you a sense of the reproducibility of the data. But suffice to say, for the purposes of this talk, that mutants in clusters, in the orange cluster, the blue, and the brown cluster here, include mutants that are highly attenuated for pathogenicity in whole animal tests, and also include almost all of the known regulators, bar 2. We've been able to recapitulate data that has been published. So very quickly, I want to tell you that so one of the really interesting things about this data set is that there's almost a direct correlation between fitness in these very diverse host settings. And so if host immune status is not the dominant driver of in-host fitness, then pharmacological interventions that target these fitness regulators are likely to have broadly acting relevance. And so this is highly significant, and we sought to prove this hypothesis and derive a mechanistic explanation for it. An obvious explanation was that the fitness data in the animals simply related to the basal fitness levels of the mutants, and we were able to use very sensitive quantitative analyses to demonstrate that that is not actually the case, and mutants that are slow growing and fast growing amongst those that are attenuated for pathogenicity in the mice. Cut a long story short, many of the regulators that we identified, both novel regulators and the ones that had already been characterized, are important for supporting responses to stress in the host environment. And I'm showing you a map of phenotypes here that links mutants to stresses and is coloured according to the in vivo fitness cluster that they fall into. So we're in the process now of corroborating these data in survival assays, but let me just point out that some of the most significant regulators are regulators that drive fitness to many different stresses. PAC-C is situated right here. And here's a novel regulator, 2B3, that's important for serum, acidity, high temperature and hypoxic stress that is fully attenuated in immunosuppressed animals. So overall, we've identified more than 80 novel regulators of pathogenicity, and we hope that this will focus the efforts of the research community going forwards to more rapidly approach solutions for drug discovery. I've made call outs to all of the important people throughout the talk. Just want to recognise Peter Cook and Julio Silva's help with the immunoprofiling work, and also my maths not mice team at Imperial, who helped with the mathematical modelling, as well as my PhD supervisor Herb Arst, who's coming back into the lab now as a collaborator on the new funding to help us with the inhibitor screens for pH signalling. And thank you all for your attention.
Thank you so much, Elaine. Before we get to the Q&A, I just wanted to announce that we're going to add 15 minutes to today's session. Of course, if people have to leave, we may do so, but that will allow us to have a little bit more in-depth discussion and not feel rushed. The way this works is we will alternate questions given by the chairs that are coming from the chat, or sorry, from the Q&A. Anyone who's listening, if you have questions, please continue to submit them. We will try to get to as many as possible, sometimes combining them. But if we don't, all of the questions will be given to the speakers and they can get in touch with you directly later on. Okay, with that, we will start with questions for Julian. Iliyan?
All right, so the first question for Julian: does pure Candida lysine induce succido mitochondrial signaling, as in the case of Candida albicans infection recently shown by Hovernician and al. paper?
Right, I believe those findings indicate that all Candida species seem to affect mitochondrial signaling through type 1 interferon. How the type 1 interferon signaling affects the potency or even the activity of Candida lysine is unknown. So the general fungal response and the Candida lysine response, I think, will likely be different. But saying that, I'm sure that if you added sublethal amounts of Candida lysine, that will certainly affect mitochondrial signaling, and that's something we probably need to look into.
Okay, so I will do a second question probably here.
Sure.
Okay, so the second question is related to the metalloproteases involvement in the damage response pathway. So what impact would inhibition of metalloprotease activity have on the damage pathway, and specifically may metalloprotease inhibition result in pro-inflammatory cytokine production by epithelium?
Okay, so we have done some of this and published on some of this. So if you inhibit MMPs, then that will reduce the EGFR sort of arm, the EGFR ERK arm of the pathway, not the P38 arm. And so that'll have more of an effect on GM-CSF, G-CSF, for example. So yes, these pathways will have an effect with inhibitors. I think there'll be a little effect on other cytokines because of those different pathways. So ERK and P38 seem to activate distinct pathways. So I suspect that MMPs will only target one arm, which is mainly the EGFR arm.
Thank you.
Question for Elaine. Actually, we had a couple of questions. How specific are these inhibitors to Aspergillus versus other fungi or potentially other organisms?
Yeah. Okay, so didn't have time to show the data, but we have tested the chemical fragments from the pilot screens against Candida albicans and also other Aspergillus species, and we've seen the same effect. So we are able to observe that alkaline tolerance is much decreased in the presence of those compounds. A big part of what we want to do next is to expand the screening efforts to many more fungi, so both pathogens of humans but also of plants, because the signaling pathway is very, very highly conserved. And it may be that there will be some agents that can be usefully used as fungicides that would also help with antifungal stewardship. And I think that the other important point there is that the actual sensing machinery diverges quite a lot between Cryptococcus and Candida albicans and Aspergillus. So there'll be an opportunity there to gain some clues about possible targets if we find fungi that are tolerant of some of the agents. And then a final point to make about toxicity in human cells is that, as I mentioned, we've got some inbuilt protection against that because our screen is reliant upon viability as a readout. Okay, so what we're automatically doing is circumnavigating any hits that are going to cripple the cell, and those may be highly conserved in mammalian cells. But also we're performing all of the tox studies alongside all of this. Just didn't have time to show the data today.
Okay. Julian?
All right, so this is basically a combined question. There are several questions that I'm combining in one. So a few questions related to, so different Candida isolates are known to induce differences in damage. Is that related to Candida lysine? And are there EC1 mutations common in the clinical isolates?
I would say yes, definitely. It'll depend on whether hypha formation can be maintained, because we know different strains have different ways of inducing hyphae and maintaining them. How much EEC1 is actually expressed by those hyphae, and then how much of the Candida lysine is then actually secreted. So you need all of those in place to get a proper damage response. So you could still have hyphae with a low level of EEC1, low damage quite easily. And we found loads of those strains anyway. So different strains will have a different ability to maintain hyphae expressed and secreted. So yes. And the other question?
So it's related to that basically. Are there EC1 mutations between different strains, and would that actually affect the damage?
Yeah, so
the damaging ability of those strains.
Yeah, okay, so I think, so in different sites, of course, things will work differently. So I think low-level expression will promote colonization in certain places, especially maybe the gut. So if you have less hypha formation, less Candida lysine secretion, therefore you have less damage, and therefore the host will tolerate the fungus better. So at the same time, we know that, for example, intestinal epithelial cells are far less responsive to toxin and Candida than oral epithelial cells are, for example. So it's all to do with level of hyphae, level of toxin expression, and the type of cell that responds to it. We also know that there's lots of different Candida lysine sequences in different clinical strains. And when we test the synthetic versions, they all act the same. So it all depends on how it's delivered by the strain and how much hyphae are made.
So we have a question that's potentially, I think, for both of you, inspired by the allergy story, but it's, what are your thoughts on Aspergillus and Candida being present in the lung at the same time? And in these settings, might Candida lysine potentially be less important? Might pH sensing be more important? So how do you think those two things interact?
Shall I go first?
Go on, Julian. You deal with Candida.
Yeah, so this is where I'm getting into very thin ground, not being a clinician in this way, or even studying asthma. But as far as I know, Candida and Aspergillus can be found together, for sure. I suspect the Candida part of that is probably more ignored than the Aspergillus part. If Candida lysine is being made in those conditions, then I'm sure it can probably promote or exacerbate the disease that's being mainly induced by Aspergillus, for example. So I think a lot more needs to be looked into that, to be quite frank. And we know from clinical studies, we've taken samples, especially with kids, I believe, then they're quite responsive with skin prick tests and specific IgEs to Candida, as well as Aspergillus. So there's something to be looked into there for sure.
Yeah, I mean, I think that this is such a compelling question. For a few reasons. The respiratory mucosa are so different from the other mucosa that you've spent such a long time characterizing the interactions with, Julian, and you can't help, when you work on a respiratory pathogen, notice that there's a hell of a lot we just don't know about the way that these fungi interact with that important interface. So, I mean, what we do know about Aspergillus exposures are that they're incredibly frequent. They're likely to happen at very low inocular, but continually. Rob Kramer's identified clinical isolates recently, along with Tobias Hull, that actually exhibit persistence in the lung and can promote an allergic bronchopulmonary type disease. And so the opportunities for persistence, epithelial entry, going under the radar of those enormous neutralizing immune responses we see when we chuck millions of spores at animals. I think that there's a huge amount there that remains to be discovered. But I think we're pretty sure that Aspergillus fumigatus doesn't produce anything like Candida lysine, right, Julian?
Yeah, absolutely. And just to confirm with what David Corey's found is that the mechanisms are entirely different. So Aspergillus seems to be proteases, fibrinogen, TLR4. Candida doesn't do any of that. It does a totally different mechanism through the toxin, through DKK1, for example, just as an example. So, and platelets. So I think the mechanisms are entirely different. So if you have equal amounts of Aspergillus candida and one of them is a high Candida lysine producer, then I'm sure it's going to be exacerbating something that Aspergillus is doing as well.
All right, so since we are on the topic, let's add a little bit more to the complexity here. So there are a couple of questions on that, more on the clinical side. So what is the evidence that Candida is a significant cause of allergy in people? And there is a notion here that Candida might be most relevant to adult asthmatics that are on high dose. So now we talked about Candida and Aspergillus, but Pneumocystis is the most common fungal pulmonary type response in the lung. Are there lessons from Candida story that would inform Pneumocystis?
Okay, I think you broke up a little bit there, but I think I just spotted it in the questions Q&A. First thing about the allergy in people, we just quickly mention that I think if you do skin prick test and specific IgGs on samples, they get it's a specific Candida response in addition to Aspergillus response. So I think it's I wouldn't say significant cause. I mean, I'm not a clinician. You'd have to talk to the clinicians out there, but it's certainly associated with to some degree. And on the Pneumocystis, which I'm just quickly reading that question because you broke up. I think yes. Most people, of course, are healthy, so they won't be predisposed to disease or asthma anyway. But if, for example, Pneumocystis activates similar signaling pathways to Candida, or indeed Candida Lysine through some other mechanism, then I think lessons could be learned for Pneumocystis based on even Aspergillus and Candida work. Although we do not think that Pneumocystis has a Candida Lysine-like toxic peptide, but it doesn't mean it's not producing something else, of course, that could do a similar thing.
Yeah, it's more like the link here is type 2 response, basically, between Pneumocystis and Candida. And the clinical evidence is that the Pneumocystis infections are more common in children. So there is probably, yeah, a lot to be learned.
Yeah. I don't know if Pneumocystis induces DKK1, for example.
So Elaine, there's a question about, so you've been looking at this correlation of fitness between in an immunocompetent host, but what happens if the host is immunocompromised, and how does that change what's going on?
Yeah, okay, so I think the question was about rates of clearance in immunocompetent hosts, if it's possible to have a look at that. Yeah, I've had a few minutes to think about that one. So yeah, of course it's possible to do. The challenge would be the amount of fungal biomass that's needed to do the detection, but actually because immunocompetent hosts clear Candida so effectively, it should be entirely doable, and that's neat, right? Because it gives you an opportunity to look at fungal clearance within the context of the whole tissue, and it may even be possible to link some of those events to particular cell types. There are other models that it would be really great to move the analysis into. So we've looked at 2 immunocompromised models, but it would be great to look at chronic infection, TB, prior TB, and of course also to see what's going on now in the viral pneumonia setting. We've been looking to set up some models that will allow us to do that. It's worth mentioning that there are, now that the technology is working, there are lots of things that can be done with this, and the data I spoke about today are the first generation studies, but with Matt Fisher and Mike Bromley and a team that are working to look at the problem of azole resistance in the environment and in the clinic, the Manchester team have now developed a means of barcoding clinical isolates that will allow some really wonderful studies to be made that looks at different behaviours of not mutants, but real world isolates that are causing problems in the clinic.
All right, question for Julian. So do you think that, EGFR is released by damaged cells? So what is basically the probability that this is actually affecting also neighboring cells that are not damaged by Candida? So is that releasing some type of a chain reaction on the epithelium?
We haven't looked so much specifically at that, but I would say likely yes, because cells talk to each other. If you're going to get ligands released from one cell, they're going to be targeting another cell. Type 1 interferon work that was just discussed about will have an effect as well against Candida on neighboring cells. So Yeah, as a tissue, they're going to be talking to each other all the time, and then it'll just be, the homeostatic, who's going to win, the fungus or the host, and obviously most of the time the healthy host wins, and the fungus becomes commensal. So definitely, yes, I would say there's a cell-to-cell discussion there.
Thank you.
Elaine, in the various transcriptomics works that you've done, more broad scale, and this may well be known, and I'm just not familiar with it as a non-aspergillosis person, but has GWAS identified any factors either on the fungal side or the host side that interface with this pathway that might give clues as to which of these pathway components are the most functionally dominant?
Yeah, so we can pick out lots of abiotic cues. So because the responses to hypoxia and iron deprivation and all sorts of abiotic factors are pretty well known, and in our compendiums of gene expression data, we could pull those out quite easily. What's very clear from the work that we've done is that the host environment is incredibly different to the conditions that we culture the fungus in the lab. And those differences are pretty highly conserved between the different underlying immunopathologies that we've studied. At the level of transcriptional regulation, it makes sense why responses to those abiotic factors might be, very highly conserved and relevant to both models. I think that what we're dealing there with are very broad domain regulators, but when we drill down to individual gene products, secreted proteases, etc., that are going to be causing damage in those settings, then the story is likely to be quite different. And what we've done, what we're still doing, have done, I suppose, but the data aren't ready to talk about yet, is to have made mutants in all of those secreted factors that are expressed during colonization of the lung. So that's more than 300 signal peptide encoding genes, all deleted, all tested for fitness now in both of those models. I was hoping that the data would be ready actually for the talk today, but it's coming down the track. And of course, it will be incredibly exciting to see from that data whether there are any individual factors that are dominant that may identify a fumigatus version of Candida lysine, maybe.
So question for Julian. Mucor species as well as other fungi have been shown to activate platelets. Would you hypothesize that a molecule similar to Candida lysine is involved in this activation?
Oh, that's a nice question. I would say yes. So mucor, as you probably know, secretes mucorrhizin, fantastic work from Ashraf. So that's going to be damaging cells. So if you're damaging a cell, whether it's through mucorrhizin or Candida lysine or something else, you're going to be inducing probably similar signaling pathways. And therefore, they'll all probably end up in the end product will be similar. So mucor, we have evidence potentially that they might be secreting peptide toxins, but we don't know. So we're looking into that. Or if they have a factor, or if other fungi have a factor that can bind GP1B alpha on platelets, doesn't have to be Candida lysine or a peptide, could be anything, or a von Willebrand factor protein, which is similar, then that'll activate the platelets as well. So there's lots of ways that these different fungi can probably activate and drive these kinds of systems, I would think, not just through Candida lysine.
Go ahead, Elaine.
Now I have a question for Julian. So, Julian, can Candida lysine be a target for antivirulence strategies? Are there reasons to suspect that that wouldn't work?
It's certainly something we're looking into. We're doing this together with Bernie's group. And in terms of the VHH antibodies, he's quite interested in looking at those. We're also looking at the development of aptamers, seeing if we can prevent. And preliminary data suggests that they are specific and they can inhibit the activation pathways and damage. But it is very early days. So based on that, it is quite likely that you could probably target Candida lysine and other similar kind of peptide toxins, maybe from other fungi. Some potentially therapeutically down the line, but that's, you know, way down the line at the moment. We're still trying to work out what it's actually doing and how important it is in different models. So to be discussed in the next 10 years.
All right, well, with that, we have just completed a full year of MycoTalks sessions, and I want to thank all the remaining participants. We have nearly 100 who are still here, and our speakers, past and present, my co-chair, and especially, again, the folks behind the scenes at the University of Exeter who are making this all work. A reminder to everyone listening that you do need to register for each of these talks individually. Please go to the website, sign up to receive the notification so that you can sign in and share the links that are on YouTube with all of your colleagues and friends. And with that, I wish everyone a very happy rest of day, evening, night, and that's all from us. Thank you.
Thank you.